Hi, everybody, and thank you for coming. So, as Dr. Griffey just said, my name is Marilyn Holt, and I am a curator with My Cancer Genome, and I'm really excited to present the redesign of the My Cancer Genome website today. Before we get started, I do need to note that I receive support from our development partner, Genome Oncology. However, this won't influence our discussion. All right. As you are all aware, identification of genetic abnormalities has become a really important component of cancer diagnosis, risk stratification, and treatment planning. My Cancer Genome was initially launched in 2011 by the Vanderbilt Ingram Cancer Center as the first web-based precision cancer medicine knowledge source, providing both up-to-date information on clinically relevant cancer mutations in cancer and clinical trial options for patients with selected alterations. In the initial site, all of the pages were individually written and reviewed by experts in the field, a process that could take weeks to months. As you can imagine, this made it really challenging to keep up with the advances in precision medicine. And for us, we just found that it wasn't sustainable to maintain the site with all the advances that are happening and keep everything up to date in the way that we would like. So we sat down and thought about how can we make the site more sustainable and how can we engineer it so that the site expands and advances with the field rather than lagging behind by weeks or months or possibly even years. And the solution that we came up with was to make the site scalable by templatizing it. So instead of writing pages individually, we generated templates that would automatically import data both from outside information sources such as SIFT or COSMIC, as well as from our knowledge management system, which is essentially a warehouse of manually curated assertions. This allowed the site to be scalable and really push us to the next level. So what I'm going to be doing in the rest of the talk is I'm going to first kind of go over the knowledge management system because that is really fundamental to the way that we built the site and we engineered it. And then I'm going to go over a few of our page types so that you can see the kind of information that's now available on My Cancer Genome. For the rest of the talk, I'm going to refer to this as MCG 2.0. It is the same site, though. It's at not the same site, but it is at the same link. So that's where you can find this information for yourself. So essentially, in the knowledge management system, the knowledge is imported from outside sources into the knowledge management system and then curated as necessary to generate what we call curated assertions and calculated assertions. So curated assertions are something that somebody has to go in and manually generate that assertion. So that would be things like clinical trial eligibility groups and therapeutic assertions. Calculated assertions are assertions that are calculated within the system from either curated assertions or outside sources. So that would be things like biomarker and disease prevalence. To support this system, we imported publicly available nomenclatures, hierarchies, and ontologies, which are outlined on the left side of this slide, to develop internal biomarker, disease, and drug ontologies that we manually supplement as necessary. We also import cancer clinical trial documents from external document sources like clinicaltrials.gov and manually annotate them using the biomarker, disease, and drug ontologies to generate a database of curated clinical trial eligibility groups and treatment contexts. And at this point, we have around 4,000 trials in that curated database. We also use the biomarker, disease, and drug ontologies to generate therapeutic assertions that describe the predicted response of a drug in the context of a specific disease setting and biomarker status. These assertions are developed based on drug approvals, treatment guidelines, and literature review prior to release to the My Cancer Genome website. So our pages harness these assertions, both calculated and curated, and import them into the page templates to generate a series of pages that describe both clinical trials, biomarkers, genes, diseases, and drugs, as well as pathways. And although I will say the pathway pages at this point are primarily individually developed. So that's why there's 19 of those as opposed to hundreds or even thousands of the other page types. So in this talk, with the rest of this talk, I'm going to be going through the clinical trial pages and then the biomarker pages, which are some of our most prevalent page types that we have on the site, and just kind of lead you through and show you what is now the kind of information that's now available there. So starting with the biomarker pages, each page starts with an overview section, which are primarily what we would call the calculated assertions. So this is information found from outside sources that say things like, where is the gene located? Is it predicted to be pathogenic or not in a particular variant? So I'm looking at the BRAF V600E page now. So is it, it's predicted to be deleterious. ClinVar predicts it to be pathogenic. It's in the BRAF gene. All that kind of really basic information right at the top. And then the site also uses data from the AACR Project Genie data set, which is a collection of, at this point, I want to say 60,000 patient cases, although I think we're getting a new release soon, so that number will go up. That allows us to calculate prevalence of biomarkers in specific diseases. And something that I think is really cool is that we've actually visualized this on each page for you, so that you can go to a biomarker page and actually look at a map of how is that biomarker distributed in the AACR Project Genie database. There's hover text in each of these maps so that you can actually put your cursor over each bar and find out how many cases are in the data set. And then you can download the maps for your own personal use. We also have sidebar links that are meant to help users quickly navigate to related diseases and biomarkers. And then we have biomarker-directed therapy. So these are our set of therapeutic assertions, and each of these are going to be a biomarker-related therapy. So we're not including things in which, a melanoma therapeutic assertion for which no biomarker is related would have, on a melanoma page, there would have to be a biomarker requirement for that therapeutic assertion to show up on the page. So we have an overview section that sort of summarizes the set of assertions that we have, things like which drugs that this alteration is a predictive biomarker for, how many FDA approvals and NCCN guidelines that we've curated within our database, and the diseases that we have relevant associations for. And then you can open up each assertion to see first the biomarker criteria, the predicted response, so sensitivity versus resistance, the clinical setting. So that here we have the dabrafenib assertion for BRAF V600E in melanoma, and it's in the metastatic setting, but we also include our assertion sources. So if an FDA and NCCN means that it's something that the FDA indicates for use in this context, as well as the NCCN, and then MCG means that this is something that we had also identified through literature review, potentially prior to it actually being released by the FDA. And so any assertions for which you see MCG as an assertion source, those are things that we've identified via literature review or through outside sources. And then occasionally we'll have a note. And then after that, we have our clinical trials section, which basically visualizes our clinical trials data so that you can see how this biomarker is being targeted in clinical trials as a breakdown by disease, and then also by the different drugs that are being investigated in clinical trials that are enrolling patients with that particular biomarker. And something I'd really like to highlight is that at the top we have a view clinical trials. So right here, we have this view clinical trials for the alteration link, and that's where you can actually go and click on that link and then view every single clinical trial that we've curated with that alteration. One thing I will point out that we are still catching up on, and you might notice, is that we have our set of open and closed trials for drugs. That's something that's still a little bit behind since we only started curating these last year. So at this point, we have all of the recruiting and not yet recruiting trials in our database have been curated for treatment context. However, we haven't gone back and curated the closed clinical trials, or all of the closed clinical trials yet. So you'll notice that, it really seems like there weren't that many closed clinical trials. Almost all of these are open. There have been a number of closed clinical trials that we haven't curated the treatment context for, but that's something that we're getting to, and we hope to catch up by the end of the year. So, all right. And then finally at the end, we have a significance section that sort of summarizes the significance of that alteration in specific diseases. And so here you can look, get some numbers for prevalence, clinical trial summary for that specific disease, drugs that are approved to treat patients with that biomarker, and then therapies and clinical trials. And sort of just a really short brief, the succinct points for that particular disease. Moving to the clinical trial pages, so we start with an overview of the clinical trial information that has been extracted from clinicaltrials.gov. So it starts with recruiting status, phase, the URL so that you can go to that clinical trial page yourself very easily. And then we have what are curated eligibility groups, so that's disease and relevant biomarkers, as well as therapeutic context. And all of these, we try to make this as clickable as possible so that if you're interested in a disease or a biomarker or what we call an alteration group, which is a set of biomarkers, or a drug that is being investigated in that trial, you can click on the name of that disease or drug or alteration, and it'll take you directly to that page so that you can learn more for yourself. We were really trying to make this as easy as possible to fall into a wiki-style hole of just investigation and learning. If any of you might remember that. And then, of course, we also have the clinical trial document extracted from clinicaltrials.gov, so that you can read that for yourself. But then, of course, you can also go to the website. That's about it. I'd really like to acknowledge the rest of the team who have worked very, very hard on this, my cancer genome team. And of course, we also are our development partner, Genome Oncology, as well as our other funders, which include the Susan G. Komen Foundation, Edward P. Evans Foundation, the Joyce Family Foundation, Robert J. Kleinberg, et cetera, et cetera. You can read those. And if you have any questions, I'm here, and also after coffee breaks, please feel free to come up and ask.
This does work. We do have time for questions. Does anyone have any questions?
Hi there. This is a wonderful resource. If we find a My Cancer Genome page, you can pretty much take it to the bank. It's wonderfully high quality. So many of us have got custom homebrew reporting software, and we have these lists of variants, and we live in fear that we're going to miss a clinically significant one. Mm-hmm. I was just looking on your website there, and it mentions API access might be a thing. You can make a programmatic call to your resource and return something back. I was wondering if you could tell us a bit more about that and maybe the possibility of connecting our resources to yours.
Right. So that part where I said that my connection with Genome Oncology would not influence this discussion, it's going to influence it a little bit here now because that API is available through Genome Oncology. And what you would be, that is available, I can give you the information for the person to talk to there, that they're the experts on that. What I can say is that what you would be accessing in that case is the knowledge management system. So that curated resource of our clinical trials and then our assertions, and we actually have there's a lot more to it than what I've shown since I've been really wanting to focus on what's directly relevant for the website. But yeah, that's something that's definitely possible, and you would want to get in touch with them for that information. So.
Any other questions? Beth?
Yeah, Beth Patel, Mayo Clinic. I'm just wondering, how often are your clinical trial resources updated on My Cancer Genome?
Right. So I would anticipate monthly because there's a two-step process. So I'm the primary curator of clinical trials at this point, and so I go through every day and I curate all of the US recruiting and not yet recruiting trials. So the US is kept up. So we have those in our system. However, then they have to go through the review process because clinical trial documents can be kind of complicated, and even when you have a lot of experience, it's easy to miss something or make an error. So we always have another set of eyes, a trained set of eyes, Neha, actually, who's about to give the talk right after mine, who goes through and just double checks to make sure that we got it exactly right. And going through that process can take up to about a month, but we really do try to keep on top of it and just make sure that we constantly have things coming in. So, yeah.